Initially, we start to work on ribosome crystallography in Russia. And then we went to United States. And currently, we are working last 10 years, more than 10 years, we are working in Strasbourg in France. So I'm going to show you results which we step by step obtained, and I summarize them in this presentation. I summarize as a questions about messenger RNA in translation. So I'm going to speak in a structural biology field and only selected the data which we obtained by X-ray analysis. And it will create some more questions, I suppose. But everything what we are doing based on biochemistry. And I'm going to show it each time when I will show the project and question. Every time it is based on the biochemistry and X-ray structure analysis answer for the question. So first, I just will remind you the information about function of ribosome. So if you remember that the ribosome is responsible for protein biosynthesis in any cell, so it uses messenger RNA on the cell, and it initiates the translation when the ribosome finds the start codon located in a 5' end of messenger RNA. This step has name initiation of translation. Then during elongation, this is cycling. The ribosome incorporates in each cycle one amino acid in the synthesized protein according to the sequence which is written in messenger RNA. This step has name elongation. And then ribosome ends the work on the stop codon of messenger RNA. This step has name termination and recycling. After stop codon, ribosome dissociate from messenger RNA and can start initiation of translation again and produce the same type of protein again. So this is kind of basic thing, but I'm going to focus now in elongation step because this is real protein synthesis itself. We are going to speak only about one cycle, incorporation of one amino acid in a growing peptide. So we simplify everything. So we have ribosome, messenger RNA, and 3 tRNA binding sites which are located in interface area of ribosome between 2 ribosomal subunits. This is small ribosomal subunit and the large ribosomal subunit. The tRNA binding site, aminoacyl tRNA binding site, A site, peptidyl tRNA binding site, P site, and exit site of the tRNA. After deacylation, it leaves the ribosome from that site. So here we have tRNA with the peptide on it. So the new amino acid comes to the ribosome with delivered by the transfer RNA in a complex with elongation factor TU, which is G protein. And this complex binds to A site of the ribosome. And after GTP hydrolysis, TU factor leaves, and tRNA can go for accommodation. It's changed orientation, and now the new amino acid which came with the tRNA located on the 50th on the large ribosomal subunits very close to the peptide of the tRNA in P site. After this step, we have spontaneous peptide bond formation. And now we have the peptide on the tRNA in A site, and the P site tRNA become deacylated. In order to continue the process, all these ligands, they have to move in one direction. A tRNA has to move to P site, and deacylated tRNA moves to exiting site. And this process catalyzed by second factor, and this is protein factor G, which is also G protein, binds to ribosome and helps in translocation. By this translocation, tRNAs move, and simultaneously messenger RNA moves three nucleotides from one side to another. So all these processes are very precise, and it's very important. Do not do any mistakes, especially frame shifts, when the location of tRNA has to be very stable and strongly oriented from one side to another. So now we practically have initial situation when I start to speak about it, and we just have the tRNA in P site plus one amino acid. So like that, we have a process of protein synthesis, and this is normal, well known, well described in any textbooks how ribosome works. But this is scheme. And what we are going to talk today about, and what we are our goals in our group during years already, is to describe this in three-dimensional manner, to show everything in a three-dimensional space, and to describe the processes on the chemical level, when you can describe every reaction as a chemical reaction. So of course, of course, when we start to work on this material, and not only we, but we are approximately 5-6 laboratories in the world who are working in ribosome crystallography. Of course, we are friends, and we are competing between each other, so I don't want to say that everything produced by our group, but I'm mostly focused on the data which our group produces, and this is about messenger RNA and tRNA, also mRNA tRNA interactions. So because we are structural biology people, we minimized the knowledge of the ribosome to the simple thing. Ribosome is only 2 subunits. Messenger RNA is a short fragment which is mimicking the real big messenger RNA. No extensions in a 5' and 3' end. And tRNAs are normal tRNA. Structure was solved much earlier. So, and now we have three-dimensional structure of this, not any more schemes, and everything what we are trying to explain is, as I said, that it is X-ray data. In the beginning of all this story, the first question was very important. Even if we know general structure of the ribosome, the major ligands are very important. Location of these ligands, and these ligands are messenger RNA and tRNAs. Because knowing the location of these ligands on the ribosome, we can somehow imagine the whole mechanism of protein biosynthesis. So I'm going to speak about messenger RNA. And here, as I said, that many things what we did in the structure investigation based on the facts which we knew from biochemistry studies. For example, one example is here. We knew that the ribosome in the cell-free system, just outside of cells, when there is only pure ribosome, 2 factors, G and TU, and amino-acylated tRNA without messenger RNA can synthesize the protein. And the protein will be just statistic. So the ribosome has this ability to produce protein without messenger RNA, without this sequence, which is of course important. So it is shown here, and it is actually a PhD, one of the major results of PhD program of my wife. She is here, and Yusupova Gulnara. And long time ago, so they show that in the cell-free system, ribosome can produce polylysine peptide. As you can see, that if you have complete systems containing TU factor and G factor, in this case it produces. But if you remove one of them, there is no protein synthesis. Then they identified the product, and they show that in the presence of all compounds, the lysine can be 2 lysines, 3, and until 7 lysines in the mixture. So it means that it's a real production of the protein. And if you remove one of these factors, like elongation factor G, the protein synthesis stops on the dipeptide formation. A dipeptide formation is quite simple. It's just 2 tRNAs on the ribosome, both of them amino acylated, and they can form dipeptide. So knowing this, we created experimental model for X-ray analysis. One ribosome contains full ribosome, the tRNA in P site and in E site, and messenger RNA fragment. Another model has everything the same except no messenger RNA. So we knew that we can construct this kind of complex properly without messenger RNA. And when we calculate the electron density map, different electron density map, we can find the real messenger RNA density. So remember, in the beginning of all this business, we never had very high resolution information, and the electron density was about 5, 6 angstrom resolution. And at that moment, interpretation of such a linear molecule, which doesn't have the bump of density, was quite delicate. And so we found that density in the difference map, as I explained. And then this is the density of the linear messenger RNA from A site to 3' end, and here the piece of E site and Scheindel-Garner duplex, which is much stronger because it is the duplex. And there is a missing part is P site, which is absolutely reasonable because in both cases we have P site tRNA, so in a difference has to be 0, and it is 0. So with such kind of internal controls, for the first time, we interpreted messenger RNA path on the ribosome. So that done in the study when we were in the United States together working with Harry Noller and Jamie Kate. But it's for the first time allowed us to describe the messenger RNA path on the ribosome. Here you can see small ribosomal subunit head and the body of subunit, and the messenger RNA is lying around the neck of small ribosomal subunit. This is interface area. We can describe A site codon, P site codon, Scheindel-Garner duplex, or this is backside of the same beginning of Scheindel-Garner and the 3' end in the backside of small ribosomal subunits. It's allowed us even to describe the environment of messenger RNA and suggest that some proteins in the entrance of messenger RNA, this is 3' end of messenger RNA in the backside, surrounded by proteins S3, S4, and S5, and we thought that these guys have to play significant role in unwinding of the structure of messenger RNA, because as you can see, the message inside of the ribosome is linear, but the messenger RNA in solution, it's a structural material. It has secondary structure and very often has pseudonodes which are quite difficult to unwind. So and just to finalize that we finally were able to mark position of plus from minus 12 nucleotides to plus 12 nucleotides area of messenger RNA, which is stabilized by ribosome, and actually it's only the type only the part of messenger RNA which is visible for us, because the rest, if it is flexible, it's become transparent for X-ray analysis. So now we had some kind of tool to study messenger RNA interaction of the ribosome, and we wanted to model the ribosome at different states of translation. So the first question was how ribosome interact with messenger, how ribosome interact primarily with messenger RNA. we know in bacteria the very important thing is Shine-Dalgarno duplex formation. And we thought, it is probably correct until now, that the messenger RNA comes to the ribosome and forms first of all duplex with Shine-Dalgarno duplex. Shine-Dalgarno duplex is 3' end of ribosomal RNA and 5' end of messenger RNA. Because like that, we can get very strong orientation of messenger RNA on the ribosome where the P codon will be in P codon without any shift in the right or left, and like that the ribosome will start correctly protein synthesis. So when we synthesized the very short analog of Shine-Dalgarno and immediately got Shine-Dalgarno density, and this is the model of Shine-Dalgarno duplex on the ribosome. And we found that the end of this Shine-Dalgarno duplex touching the protein S2, which located actually in the backside of the small ribosomal subunit. This is kind of marker. Then we created two types of complexes. And the difference between these two types of complexes, one has named initiation complex, where distance between AUG start codon and the adenosine of Shine-Dalgarno sequence in 7 nucleotides long. And this statistically normal distance for most part of messenger RNAs. So when we form this complex and we solve the structure, and we found that this is the location of Shine-Dalgarno duplex is different from what we found with a small fragment. It doesn't touch the protein S2. It has a far distance. So then we created another type of complex, which I show here, where we give the ribosome to choose position of the start codon just by putting UUU sequence here, which is in any orientation, it's a coding only phenylalanine tRNA. And like that, ribosome automatically choose position of the start codon in a distance 10 nucleotides. And when we solve the structure of this complex, we found, this is the first one we name initiation complex, and in the second position was like beginning of translation. So we named this relax complex, and the first one initiation, tense complex. Yeah, and finally we also wanted to see the situation when translation already starts and there is no more Shine-Dalgarno, and what part of messenger RNA is interacting with ribosome, and we solved the structure showing that the ribosome interacts with messenger RNA from position -5 to position +12. So now we can summarize everything and describe like a scheme of interaction. So messenger RNA comes to the ribosome, form Shine-Dalgarno duplex, and Shine-Dalgarno duplex located very close to protein S2, which location is actually not in the step of initiation. It's location on the step of elongation when the complex is relaxed. So it means that in order to bind tRNA in P site in this initiation complex, the whole messenger RNA has to move a little bit back and then fixed by tRNA in P site. And this is tense complex. So fortunately for us, that was shown before, this phenomenon shown before by Claudio Gualerzi laboratory here in Camerino in Italy, and they show that this phenomenon exists, and they show it by cross-linking experiments. So it was very supportive material for us that we show not something very artificial. It is not artificial, it shows like this. So binding, then adaptation of messenger RNA, and then beginning of translation. So, and then finally we describe a scheme like this way. So slowly we move to higher resolution. And by high resolution, the many things become visible. Not only density of messenger RNA, but also some new elements. For example, we created, so now we can describe all interactions of messenger RNA on the ribosome. This is the slice. You can see here the slice of ribosome on the level of messenger RNA. This is the entrance of messenger RNA. Then it's some kind of downstream tunnel, the hole where messenger RNA goes to A site, form codon-anticodon interactions with A site, P site, E site, and then goes out, and this is Shine-Dalgarno helix interactions. So, and we describe this type of complexes now at chemical level, at atomic resolution. And for the model which we study, we use elongated complex, as I explained you before, when the messenger RNA has UUUU sequence, which in formation of complex we have 3 tRNAs, phenylalanine tRNAs on the ribosome. And what was interesting, phenylalanine tRNA has anticodon AAG, which is according to the codon UUU, but in the front of anticodon there is adenosine 37, which has modification, which is the modification, the presence of modifications in position 37 is common for tRNAs. But for specifically, and very often, this modification is like in phenylalanine case. Here you can see methyl TO group and one additional element which we never seen the importance of this element in our study. But methyl TO group play very important role. Here you can see interactions of codon-anticodon duplex of Phe tRNA and UUU codon in A site and in P site. In both cases, in the front, you can see adenosine 37, which has this TO group modification, which form very strong stacking with the first base pair of the codon in A site and in P site. Again, coming back to biochemistry. Biochemically is known by people who study these modifications many years that the binding of the tRNA to A site, Phe tRNA in A site, without modification and with modification, very different. In case of presence of modification, the interaction and binding much stronger. And now we can explain it why. It's just because of this additional stacking interaction. But much more interesting for us was to see codon-anticodon interaction in E site, because that was the big discussion in the world of ribosome, presence or absence codon-anticodon interaction in E site. And we see very strong interactions of first base pair in E site, and it's supporting also again by methyl TO group with adenosine 37 in the front. But next pair, like second base pair, has already distance 3.7 angstrom, which is higher than Watson-Crick interaction, Watson-Crick distances. So probably there is no here. And in the third base pair, there is no base pair interaction. More than that, because of the high resolution data, we found there is one magnesium atom very close to this modification, and this magnesium atom completely, fully with water, hydrated, sorry. My English is so fully hydrated magnesium atom, and it forms very interesting net of interaction between this modification between ribosomal RNA elements and the messenger RNA, the first codon of E, the first nucleotide of codon of E site. Without this modification, no net of interaction, and that explains another type of biochemistry, which is if there is no modification and there is no net of interaction of what I just showed, it is shown biochemically that the tRNA in P site is flexible, can move +1 or -1 frame shift. Having +1 or -1 frame shift, we completely destroy the product and the proteins which we synthesized like that. So that's why for the first time we explain that the major player of this is that modification and the presence of fully hydrated magnesium. I have 15 minutes more?
Yeah.
Mm-hmm. So now we are coming to one more question, which became last year quite important in our group, and not only in our group, because this still continue. It's a problem of discussion with previous data, which produced by Venki Ramakrishna laboratory, and I am going to speak right now only in the facts which we found. It's a question of decoding. Decoding problem is the major problem of protein biosynthesis, because it is transformation of one type of coding from messenger RNA to protein production, from nucleotide code to amino acid code. And how tRNAs behave and recognize, and messenger RNA tRNAs recognize each other in A site is very important question. So what is, as just to remind you that the decoding center located on the small ribosomal subunit, this is messenger RNA path, you can see here, and this is tRNA in A site, and but peptidyl transferase center located 70 angstrom away on the large ribosomal subunit. But efficiency or quality of decoding are very high in protein biosynthesis, and even the protein how to say peptide bond formation and decoding are related, and the distance is very far from each other. Is it visible? I will explain. So it's the same type of thing what I show you in the beginning, how tRNA binds to the ribosome. So what we have a model now, ribosome containing messenger RNA, tRNA in P site, and we can add tRNA in A site, changing the codon-anticodon interactions, and look how the ribosome will interact with messenger, how the tRNA will form codon-anticodon duplex on the ribosome. So tRNA comes to the ribosome in a complex with TU factor, binds to the ribosome, and after GTP hydrolysis, tRNA can accommodate and go to peptidyl transferase reaction. So the accuracy here is very high. But what is known today, that the ribosome recognizes the type of tRNA which comes to the ribosome, which comes in this step. So here I have to introduce one more terminology. So there is a canonical interaction when the cognate tRNA is coming, and there is semi-canonical interaction where neocognate tRNA is coming. The neocognate tRNA is the tRNA which has one wobble pair in the codon-anticodon duplex. I will show you later. But ribosome recognizes the differences. For some reason, ribosome accepts with a high efficiency the correct tRNA, cognate tRNA, and then it goes to peptide bond formation, and ribosome can refuse tRNA if the tRNA is neocognate. And this step has name proofreading. So first, because we are structure people, so first we look inside and we can describe what kind of ribosomal elements are important for recognition of codon-anticodon duplex. It is shown by Harry-Noller laboratory by chemical modifications that there are adenosine 1493, adenosine 1492, and guanosine 530 involved in stabilization of codon-anticodon duplex on the ribosome. It was shown before. Then we can see also by Venki Ramakrishna using ribosomal small subunit and simulated elements like small messenger RNA and anticodon stem loops. They show that the orientation of these three important nucleotides depends on the presence of tRNA. Without tRNA, these two nucleotides inside of helix 44, but in the presence of ligand, they flips out from the helix 44 and they have another orientation. And like that, they are involved in stabilization of codon-anticodon duplex. I will show you how. This is also data from Venki Ramakrishna laboratory. Now it's classical that 1493, adenosine 1493 stabilized first base pair in codon-anticodon duplex. Adenosine 1492 and guanosine 330 stabilized the second base pair in a codon-anticodon duplex. So we created, for comparison, control complexes, which is normal cognate tRNA with cognate codon, and two type of necognate tRNAs, where I say that the one base pair should be wobble. Here we introduce GU base pair in the first position, and here GU base pair in the second position. So when we solved the structure of the ribosome, we found very interesting phenomena. The geometry of GU base pair looks no difference with classical GC base pair. So GC base pair is the classical Watson-Crick base pair, Watson-Crick geometry base pair. And GU normally, from our knowledge, should behave as a standard wobble base pair, which shown here. The wobble base pair has a shift. Here, by analyzing the density of electron density in the ribosome, the GU base pair in the first position has the same geometry as GC base pair. When we look at the second complex, when we have GU base pair in the second position with stabilization, as you remember, these two nucleotides, it has exactly the same geometry like AU base pair, again, like a classical geometry, which is kind of different point of view than it was before. Because before we thought that, according to Venki Ramakrishna studies, we thought that ribosome recognized the Watson-Crick geometry, wobble geometry of the tRNA. But looks like in our case, ribosome force wobble to take the geometry of classical geometry of Watson-Crick. And only in the third position, which is not important for decoding, in the third position, the geometry of GU base pair is classical standard wobble. It's much flexible there. So in the two first position and the second position, the pocket which ribosome forms are so restricted. So it's accept only Watson-Crick geometry. So that's why Watson-Crick, that's why wobble pair behave as a Watson-Crick. How it's possible? It actually was predicted again by Watson-Crick that it is possible if the form if the interaction is not keto interaction, if it is enol interaction, it will have the same type of geometry like keto for GC. You can see here GU geometry has the same geometry as GC keto. This is tautomeric shift of the nucleotides, which is theoretically possible, and it was predicted by Watson-Crick as a possible mechanism of mutation in DNA duplication. So this we have in our case RNA, and it is actually also shown by RNA polymerase in RNA. No, it was in DNA polymerases cases that is possible. The incorporation of GU as a normal base pair according to the rules of Watson-Crick. Again, just to show you, to prove what I said, that these three nucleotides which are actually organizing the environment of decoding center, these nucleotides are not flexible. And it was important to show. So we show in case of cognate tRNA complexes, this is the position of these three nucleotides, what I described before. And then we solve the structure of neocognate tRNA versions, and you can see that there is no practically difference in orientation of these nucleotides. So it means that it's really rigid pocket for two first nucleotides what we have in a complex. At the same time, we know that the binding of tRNA in a site changed the structure of small ribosomal subunit. You can see here comparison of ribosomal RNA structure in the presence or absence the tRNA in a site. But it happened the same way for cognate tRNA and neocognate tRNA, and even non-cognate tRNA. If we are able to put it in a site, it will have the same type of changes. Another thing which was very important for us is to explain not only the ribosome part of interaction with the duplex, but also messenger RNA part. And the messenger RNA part is extremely restricted, also very rigid. It's because between two tRNAs in A site and P site, in order to have them in parallel orientation, the messenger RNA has to make a kink. And this kink, very stable and actually stabilized by ribosomal RNA element, that's cytosine 1400, and 3 magnesium atoms around. So it's also very rigid part of interaction of this rigid part of the environment of codon-anticodon duplex. That's only for comparison that we found the differences between cognate and neocognate tRNA in the structure of anticodon stem loop. We don't know, is it significant differences or not significant differences, but maybe if it is significant, maybe this is the way why ribosome reject the tRNA from A site, if these differences are so significant. Also, all the structures which we analyze in the presence or absence of paramomycin. This antibiotic shown before as very important antibiotic to decrease the restriction of binding of neocognate tRNA. Yes, it's possible to use this scheme for explanation that the presence of antibiotic increase a little bit size of tRNA, codon-anticodon pocket. And like that, incorporation of neocognate tRNA will be easier. it's less important. It's awesome. Yeah. I'm going to finish my talk. I just wanted to say that this part of work was what I was discussing during all the time. As I said, that in the beginning, the first part of research done in University of Santa Cruz in California with Harry Noller and Jamie Kate, and my wife and Marat Yusupov was involved in all different type of complexes and different type of projects which presented here. And yeah, I'm sorry. Lasse Jenner was, he's not here anymore. He's professor now, so he left our laboratory. Sorry, this is not the right slide. Yeah, and yeah, Lasse Jenner is the crystallographer and responsible for the last projects which we made. And Natalia Demeshkina was involved in interpretation of decoding process, which I presented at the end of my talk. Thank you very much.